Hello, in this video we will discuss about the aspergillosis. This is a fungal disease. Basically the aspergillosis which that is caused the multiple type of diseases due to the fungal attack. An infection due to species Aspergillus. This is a species of the fungi. Due to the species Aspergillus to cause the aspergillosis. So let's begin to understand the mechanism of infection, and we will also classify of aspergillosis with the overview. So this is the hyphae are present in the soil will produce the conidia, and conidia will move into the airborne conidia into the air. When go into the air, it will move into the trachea and lungs. After going to the lungs, the allergic reaction will clear this process through an innate immune response. But sometimes the primary clearance escape this rodlet cover of the spores, and the infection will progress due to sometimes this is cleared through our innate immune system and the macrophage are present in the lungs, but the weak immunity which that will lead to cause the aspergillosis. This is the alveoli contain epithelium, the pulmonary epithelium and the blood circulatory system. So after this, here is a rodlet cover containing the spores. This is known as conidiospore. And the primary clearance escape number 1 will lead to bind their residue, the sialic acid residue and lactin acid residue will bind with a specific receptor are present in the pulmonary epithelium if the macrophage will not work, if the immunity will be weak, or the HIV disease. So on the other hand, the pulmonary epithelium here and the fibronectin receptor, fucose, residue of N-glycan, and the melanin containing coat are present in the spore. So oxidative stress inhibition by this. In this way, the oxidative stress will also inhibit the immune response. So this is the sialic acid residue and lactic acid residue bind, and the pathogen recognition receptor also bind for the invasion. So remember that things. Basically, this is a resting conidia still. But in the case of swollen conidia, after the binding with the dactin 1 and the CALA. So the CALA and the pathogen recognition receptor bind. After this binding, the CALA and the beta-1,3 glucan. This beta-1,3 glucan and CLA will collectively bind for the invasion with the pathogen recognition receptor, which that will recognize the pathogenicity and invade. So the rodlet cover and melanin shed, this melanin shedding and rodlet cover will be shed will lead to invasion. So here is the dactin, and this is the beta-1 integrin, yes, beta-1 integrin will bind with the spores, and in this way the internalization will trigger, and by this here is you can see the dactin will activate the PLD, will lead to phosphorylation, will lead to activate, and in this way the internalization will occur, and the pathogen recognition receptor will activate the cofflin 1, which that will release the free actin, which that will be bind after the phosphorylation, and this actin is used for the internalization of this pathogen, this spore. After this germination, conidia will occur by the invasion. So basically the various pathogen associated molecular patterns we recognize in this manner. So remember that things. After this is the endosome formation will occur. This endosome formation and the dactin basically inhibit by this, and evasion and germination will occur. In this way, the germination which that contain beta 1,3 glucane, either it go or go in the evasion and germination, or phagolysosome. Phagolysosome is very important, will clear the pathogenicity, while the glycosaminoglycan will lead to adherence of these spores. So remember that things. Anyhow, the destruction will occur after the germination conidia, the destruction of the epithelium. Remember that things. So in this way, the glycosaminoglycan, which that will trigger the fumagillin, this fumagillin and glucosaminoglycan, and secrete the fumagillin and proteases, and this will lead to tissue degradation. And in this way, the basically the apoptosis occur due to the fumagillin, and the fumagillin basically apoptosis, and on the other hand the glucose aminoglycan, which that will lead to natosis, and interleukin and IRA, which that will trigger the pro-inflammation and the tissue degradation due to the fumagillin also. So anyway, this is the process generally I am describing about that, but you can just understand about that. This will lead to basically the apoptosis will lead to immune response inhibition, because apoptosis is occurring. So in this way, I hope you make sense about these things. Trigger the apoptosis and inhibit the natosis. Sorry, so that is why inhibition of the natosis. The neutrophil will not use for it. So remember that thing. So in this way, the immune response inhibition due to the apoptosis of the epithelial cell. The pulmonary epithelium will lead to cause the degradation and invasion into the blood circulatory system, will uh, cause a different tissue infection, the infection progression, the different for example lungs, liver, stomach, large intestine, small intestine, every type, for example brain also. So let's begin to understand the classification of Aspergillus. Classification of Aspergillus is the Aspergillosis, which that will lead to invasive, the angio-invasive sinusitis, basically the invasive Aspergillosis, pneumomycosis, systemic aspergillosis, and aspergilloma, and allergic aspergillosis, which that will activate the mast cell, will lead to cause allergic reaction. But this all classification is through a aspergillus species. So anyhow, this is the pulmonary mycosis to pneumomycosis, and on the other hand, the invasive aspergillosis through will lead to cause the angio-invasive sinusitis, colonic, pulmonary, rhinoorbital, tracheobrachial, and the putative invasive pulmonary, chronic invasive pulmonary, invasive sinonasal, and granulomatous invasive, and the rhinosinusitis. This is the all type is the invasive. Invasive mean the invasion of the spores after the germination. This will lead to after the germination to invade the several organs to cause this type of disease. While the systemic mean the pulmonary, ocular, and the urethral, cardiac, GID, central nervous system, and renal, cutaneous, and disseminated and others. while the aspergilloma is the pleuropulmonary, and the pulmonary will cause the aspergilloma. And allergic aspergillosis due to the allergic bronchopulmonary and allergic pulmonary aspergillosis and the cyanobrachial allergic allergy. It is the type of the allergic reaction. So this is the aspergillosis which that will lead to progress to cause several type of tissue damage, and the damage of the tissue will lead to cause the severe infection. This severe infection due to the fungi aspergillus due to the inhalation of the spores. So I hope you make sense about these things. This video was about the aspergillosis, the fungal disease, and the fungus is the fungus is nor plant, neither animal, but it is containing a cell wall with chitin like arthropod. So thanks for watching. Please make sure to subscribe.